Hello, my name is Eleftheria Bakhchevani and I'm going to show you recent results we have regarding the on-target and off-target effects of DMPP on the soil microbial community. DMPP is a leading nitrification inhibitor in the global market, which according to a recent in vitro observation of our group, shows a selective activity against soil ammonia-oxidizing bacteria, while its effect on off-target soil microbes remain unknown. Here, we evaluated the impact of DMPP applied at two dose rates, a low one equivalent to the agriculturally recommended application and a high dose aiming at universal ammonia-oxidizers inhibition on the function, diversity and dynamics of ammonia-oxidizers, nitrite-oxidizing bacteria and denitrifiers, and the overall prokaryotic and fungal soil community in two soils of similar physicochemical characteristics but contrasted pH and microbial community structure. In parallel, we monitor the dissipation of DMPP in soil to provide an estimate of the level and the duration of soil microbial exposure to the NI. DMPP showed a dose-dependent dissipation pattern and higher persistence in the acidic soil. DMPP did not appear to impose an accumulation of ammonium in the two soils. In contrast, its low dose induced a significant and persistent reduction of potential nitrification and nitrate concentrations, mainly in the alkaline soil, and only the high DMPP rate uniformly inhibited the conversion of ammonium to nitrate in both soils. This higher activity of DMPP in the alkaline soil, despite its lower persistence in this soil, was most probably driven by its higher activity towards AOB prevailing in the alkaline soil as verified by qPCR and RT-qPCR, unlike AOA, whose abundance and transcriptional activity was reduced only by the high dose. Amplicon sequencing revealed significant effect of DMPP on the diversity of AOB communities in both soils, unlike AOA, which were less responsive. Changes in the community of AOB in both soils mostly reflected in the relative abundance of members of genus Nitrozospira, particularly Nitrozospira sp and sp12, which were suppressed by DMPP in the acidic soil, and members of the cluster 3b Nitrozospira briensis in the alkaline soil, which were either suppressed or favored by DMPP. AOA community composition was significantly altered by DMPP only in the alkaline soil, with DMPP imposing varying effects on different Nitrozospirales clades. Nitrozospirales delta 1.2.1.1 clade was negatively affected by DMPP, unlike ASVs belonging to gamma 1.2 clade, which were favored by DMPP. Beyond on-target effects, DMPP at the recommended dose rate did not show a consistent effect on the abundance of NOB and denitrifying bacteria, although we noted for the first time a selective inhibition of Nitrobacter but not Nitrospira by DMPP at a more than 100 times higher dose rate. The application of DMPP induced significant structural changes in the communities of bacteria and fungi, with the fungal community being more responsive to the application of the NI. Particularly, we noted a clear negative effect of DMPP on bacteria of genus Nocardioides in the acidic soil and of the genus Lysobacter in the alkaline soil, and the stimulation of Sphigomonas in acidic soil. Regarding fungi, DMPP negatively affected the abundance of fungal genera like Exophiala, Trichoderma and Lectera in the acidic soil, and Ascobolus and Metacordyceps in the alkaline soil, while stimulated Tetracladium in the acidic soil, Solicocozyme in alkaline soil, and Fusarium and Coniochiata in both soils. These results suggest that DMPP, especially at higher concentrations than the recommended, is expected to affect key soil microbial functions beyond ammonia-oxidation, threatening the soil plant ecosystem homeostasis. Overall, our study expands our understanding of the interaction of NIs with soil microbial community beyond target organisms and paves the way for a more comprehensive analysis of effects of NIs on the soil microbial community. Thank you.